So I had asked you this question. Did anyone has any ideas or any guesses? Yeah, right, right. Okay, post Okay, okay, that's good. But this one is much simpler, right? It's like I'm asking if you had um, for this task, what could you have done, right? So I will, so with, I'm saying this because with Bart, I have maybe 15 papers using resting state with his task to look at different aspects, right? So I'm going to discuss maybe only 5 or 6 one by one, and how, like, make you think how we can use it, right? So the first one was hemodynamic scaling of fMRI bolt signal, validation of low frequency spectral amplitude as a scaling factor, right? And in this, what we show is that this is time course of representative voxel from the motor cortex in a typical subject during finger tapping, right? So this is finger tapping. B is breath holding. C is moderate hypercapnia, right? So these are 3 different paradigms, right? This is finger tapping, this is breath holding, this is hypercapnia. And what this shows is the relationship in change between hypercapnia, which is 5% CO2 versus breath hold, right? And so on a voxel-wise basis, there is a very good correspondence between breath hold and CO2. And in some way, it is not very surprising because for both the stimulus, there is going to be an increase in blood flow, right? Both are vasodilator, right? And both of them, there will be increase in blood flow. So the voxel where there is an increase is also going to have an increase, right? And here you can see the change, percent change can be as high as, 50% or even higher, right? So this is percent change during breath holding to the ALFF, or the magnitude of the frequency spectrum during finger tapping, right? So what I'm doing is I'm doing a FFT of this time series and comparing with the percent change, and showing a good correlation, like not a very high correlation, but nonetheless decent correlation, right? So this is breath hold, square root of finger tapping. This is breath hold also with the square root of. So for different subject, we see these changes. And so this, um, it doesn't look as clear. So this is the scaled version, sorry, the unscaled version. When I say unscaled version, it means that I'm simply plotting the response for motor task for subject 1, subject 2, and subject 3. This color represent, red is more active, right, between 0 and 10. So I'm mostly plotting between, let's say, 5 to 10%. So this is without doing any scaling. Again, remember that breath holding is the gold standard, or 5% CO2 is the gold standard to study CBF, right? Because that is only flow information without any neuronal changes. And so this is using the breath holding to scale the response. And this is using the resting state Fourier spectrum to look at these changes. And this is the distribution across different subject, right? The bold during finger tapping, right? This is the bold change. This is different subjects. And what we show here is the bold change, again, for different subject. But here we are scaling the information, right? So this is unscaled. This is blue is scaled by breath holding, and red is scaled using low frequency spontaneous oscillation, the amplitude of the low frequency signal, which is our resting state signal, right? And so the distribution of these signal changes looks so similar between breath hold and um, resting state, right? So this is ways in which we can we can calibrate our data, right? This is again percent change in bold during finger tapping, percent change in bold during breath hold, right? Again, this suggests that the change, the amplitude of the magnitude of the change that we see during task, there is a large hemodynamic component, voxels which have good changes, which will change a lot during breath hold, is also going to change during finger tapping. And so if you simply plot your response, then what you are seeing is certainly activation, but that is affected by the hemodynamics, the magnitude of the hemodynamics, right? So these are the different subjects. And the point is that we can use this to calibrate the data, right? So this is another study. The title is Neural and Vascular Variability in the fMRI bold response in normal aging. So this is the way we plotted this is we had 11 subjects, and here we are asking there should be a voxel is active if in at least 5 of the subjects there should be activation. This is a voxel is active if in at least 6 of the subject that voxel should be active, like in 7, 8, 9, okay? And the same for older subject. So if you look carefully, what you see is that this is finger tapping. This is finger tapping for young subject. This is finger tapping for old subject. So what do you see from this figure? What do you see? Do you see any trend? Say something. Michael. Yeah. Okay, good. So what Michael is saying is that for the young subject, between this map and this map, there is variation, but not a lot. But in the older subject, between this map and this map, there is a lot of variation, which would suggest that from in older subject, from subject to subject, there's a lot of variable, right? And as you look at overlap, it decreases significantly, right? So here, of the 11 subject, at least 5 subject is active. This is at least 6 subject, 7, 8, 9. There is a significant reduction. In here, this is young subject. There is reduction, but not as much as young subject. This is another task called digit symbol subtraction test. It's substitution test. And this is like plotting the same way, right? This is 5, 6, 7, 8, 9. There is not much overlap here. First, it is not as, like the area is not as big as motor, but we do see voxels here, right? But here there is already a reduction, and then further reduction, barely anything left, right? So then the question was, what could be causing this reduction, right? Is it all neuronal, or is there a vascular effect? Okay, and so what we did was we had also done a breath holding task, right? So this is the response compared for young subject, and this is the response in older subject, right? And what it shows is that in older subject, there is much less activation, right? Again, that would suggest in older subject, overall, the response is less, right? I'm not saying this explains everything, that all the decrease is due to the lower hemodynamic response function, but certainly some part of this variability is due to this. Okay, is that clear? Again, ask me if you have any question. So again, this is yet another paper. This is non-neural bold variability in block and event-related paradigm. This is the block design. This is the event-related design. And basically what we are doing is we are calibrating the responses like we did in the previous one for both block design and event-related design, and showing that it can be used for both block design and event-related design. Because in this case, my friend Bart, he thought maybe event-related already accounts for the hemodynamic response function, and maybe we would not need to do the scaling. So that is why we had done the study. So the other study we did was, so this is increasing measurement accuracy of age-related bold signal changes, minimizing vascular contribution by resting state. fluctuation of amplitude scaling. I would say this is where finding a good acronym is also important, right? Um, so, so this one is showing changes in young subject and older subject. And here what we show is, this is, right? So this is motor, this is cognitive, right? Right? During task, younger, older subject, right? A voxel-wise cross-correlation of the bold signal time course was performed with the reference function representing the task paradigm for each subject convolved with a gamma variate function. And we see differences in activation pattern. And again, in this case, like this has been right, during cognitive task, a voxel by bold was performed. Again, this we are using standard analysis and showing that between young subject and older subject, there is a difference, and that by using, right, this is young, then RSFA for the young and RSFA for the old. So after we do the scaling, what we find is that the difference in the distribution, which was here, is minimized, right? Also, the width of the distribution is also significantly reduced, right? Again, motor, motor, cognitive, cognitive, right? Again, I'm not saying that this is not important, the distribution is not important, but what I'm saying is that in many cases you can calibrate to see if there is any differences, right? The fact that these two distributions are so similar suggests that maybe for motor task there isn't going to be a large difference, right? This one is, again, prediction of task-related bold fMRI with amplitude signatures of resting state fMRI, right? In this case, what we do is, these are the maps of activation, right? Structure from the motor region, spatial and temporal, right? A is resting state, B is motor task, and C is breath hold task, right? And what this shows is that if once we have identified the regions of activation, if we then display the percent change or the standard deviation, they look very similar, right? So this is motor, this is breath holding, right? So here, look at the red spots. So the red indicate large activation in those regions. The green, intermediate, right? Look at the breath hold response. This to this, this to this, this to this, right? Right? So again, suggesting that during breath holding, the regions where there is large change during breath hold, those regions also there is large change. Okay. Now, if we look at the resting state variation, we also observe that the voxels where resting state has large change is also the same region where task and breath hold have changed. Again, it's not a complete one-to-one correspondence, but nonetheless the map look very similar, right? So like this one, this one, this one, this one, not so big, but it is there, and this one, right? This, right? This. So typically voxels which have large change during task, you should be careful before saying that they are more active, because those voxels most likely have large hemodynamic response function, and therefore you see large changes, right? And by looking at the resting state signal, we can say that, okay, maybe we can calibrate or normalize for that effect. Again, you must remember that it's not 100% one-to-one correspondence, right? This one is active, has large change. This one has large change. For breath holding also it has large change. However, there is large change here, which we don't get in task activation, right? So it's not 100% correspondence, right? So this is breath hold change, this is motor, this is motor, this is resting state fluctuations. It is very similar to ALFF, and you see such a good correlation. Okay, that's for motor, right? This is again for motor, for resting state. This is motor, resting state, motor, right? Low frequency, high frequency. Again, we have this low and high to show that if we have the low, there is a even higher correlation compared to if we have everything else. Any questions so far? No? Okay.
Very high correlation. What are we seeing? What are we
for this one? So it for the high correlation, so this one was, this one is finger tapping, right? And I identified the regions during finger tapping, and I'm plotting how much change does those voxels have during finger tapping, right? Then what I did is, for these voxels, I wanted to see how does, during breath hold, do those voxels change, right? So this is plotting for breath hold, and this is for resting state, right? And what I want to say is that, you know, if like these, the red voxels have large change, the green has, you know, then is yellow, then is green, then blue. If I went to the same voxels during rest or breath holding, the response is also going to be similar, right? That in this region, the highest change will be in these regions, right? Similar to task activation. And this is for both breath holding as well as resting state. And the point is that breath holding is the way to measure blood flow. However, if you cannot, for some reason, if the subject is claustrophobic or subject cannot perform the task, we can use the resting state as an alternative. Okay. The next thing, the conclusion is that two things. One is when you are doing task activation, you should be careful to say which one is more active, which one is less active, right? Because the, the response may depend on the underlying neural efficiency and so on, but the response also depends on the underlying hemodynamics, right? And people typically assume that when there is large change, it is neuronal, but that is not the case. When there is large change, it could be neuronal, but it could be due to those voxels having large hemodynamic response, which it suggests. That's one point. The second point is that you should use breath holding if you want to scale the data. However, if you cannot use breath holding, resting state can be substituted and you can get very similar information. Okay. Yes. Yes, because breath holding, when you hold your breath, right, you are not taking new oxygen, so oxygen gets used up, there is more carbon dioxide, and pretty soon there is a steep increase, right? The carbon, when you breathe carbon dioxide, you get similar effect, but the effect, like you have to place a mask, right? Which adds more complication, right? Let's say here, if we wanted to do carbon dioxide, you have to get carbon dioxide, have all the setup, make sure it could be used by humans, and only then can you give, right? And even that would require a lot of safety issues. But if you are to ask someone to hold your breath, it's fairly easy. You ask them to hold it for 10 to 15 seconds, and most subjects can, right? Most young, healthy subjects. So you can do that. So the last thing I would like to show is here we are using calibration of fMRI activation based on neurovascular and anatomical constraint. So what do I mean by that, right? So let's say we have two groups of subject. You have young adults and old subject. So in older subject, is there any structural differences? Anyone? The gray matter volume is reduced very larger, right? So those are the two main issues. So then the question is, when we scan someone, older subject, is their gray matter volume? How would gray matter volume affect the fMRI signal? For task. forget about recruitment. Let's say there were two young subjects. Everything was the same, excepting one person had more gray matter volume compared to the other person. What would happen? What does having more gray matter volume mean? Anyone? Neuron also, maybe the vasculature and so on, the underlying vasculature would also be more, right? So what happened? Yeah, so you would have both neuronal firing, but also more underlying vasculature. So you would get more signal from the gray matter. And because fMRI typically has focused on the gray matter, you would get more signal. In older subject, first, I have already mentioned several times, the challenges with the hemodynamic response function, right? Not as elastic, the output is less, um, mean arterial pressure, high blood pressure, and so on may also is also going to affect. On top of that, the gray matter volume is less, right? So then if we had to do a study between young and old, can we calibrate for both anatomical differences as well as functional differences? So in this paper, we have tried to do that, right? 2 minutes. So this is showing, I'll just show. So this is showing the motor response, right? The average motor response. This is for DSST. This is for breath holding. This is for ALFF. And this is for gray matter volume, right? So we can get this on a subject by subject basis, right? And what we typically do is we get a map and say that, hey, when we do finger tapping, these are the regions that show up, right? Left and right motor cortex, SMA maybe, some in cerebellum. For the DSST, we similarly show these regions and say, okay, these are the regions which are active, right? And between healthy control, or I should say between young and old, or two different groups, there are differences. We assume everything to be related to neuronal activity or the task that we are performing, right? But this is showing breath hold, and this is showing ALFF on a voxel-wise basis. First, we can see that there is a very good correspondence between breath hold and ALFF. Not, again, not 100%, but a very good correspondence between the two. And then this is the gray matter volume. So what we did was, in this case, this is the original data. This is calibrating this response using both breath hold and gray matter volume, because these two are highly related, right? Because if the gray matter volume is going to be more, then the breath hold response is going to be also more. So that's why we used both. Also, this way we are taking into account any structural as well as functional differences due to hemodynamic. And then this is using ALFF and gray matter volume. And what I want to emphasize is that, see here, for example, this was all active, but after calibration, it's more specific. With resting state, we get similar changes, right? It's hard to say, like for this one, this was supplementary motor region. After scaling, we don't see much, but scaling using resting state, there is still some effect left. So I don't know which one is like the true way. But anyway, this is how we can calibrate the responses. Similarly, this is for DSST, right? Big changes, but after we scale it, the response is significantly reduced. And again, I'm not saying that like all this is vasculature, but vasculature does play a role, underlying hemodynamic response function. This is like, finally, this is another paper we had written looking at regional homogeneity of resting state contributes to both neurovascular and task activation variation, right? So far we had looked at like voxel-wise measures. Now we want to see that, okay, so this 4 figures you should like must know by now. This is looking at reho, like how does each voxel correlates with its neighbors. And what this shows is that, you know, when the total change is, let's say, this is the Venn diagram, relative unique and overlapping contribution of regional homogeneity, right? Amplitude of low frequency fluctuations and breath hold, right? So we can take any task and then further divide them into how much of it could be explained by like reho, how much of it could be explained by breath holding, how much of it could be explained by ALFF, and even in between, right? And by adding these up, we can explain perhaps that how much of the 100%, what fraction can be explained by breath hold ALFF and reho, right? And so once you can explain, what would be left is perhaps more neuronal, right? And this is for finger tapping. This is the DSST task positive network, and this is the DSST task negative network. So again, what I want to point out is that as you like for resting state, one way is to like make this maps, make correlation matrices, make ICA, and see how different regions talk to each other. But the other thing that you should also do is that even if you're doing task activation, like please collect a resting state data, because it will help you clean up your data. You can see how much is the underlying hemodynamic response function, you know, how is ALFF affecting your task response, how is reho affecting your task response, is the gray matter volume affecting the signal. All these things can be measured and even quantified. So it would help to maybe quantify your task response. Okay. All right. Thank you. I will be back in an hour. Okay, no problem. But don't go.